Okay, so welcome everyone, and thanks for coming to the very last lecture. So on this one, I actually spent some time today to update it, so it's really close to what we're doing at the moment. And I would like to talk about biocontrol. And the example I will be giving is rabbit biocontrol. So biocontrol is something that will happen probably more and more because it's fairly efficient and it's particularly efficient in huge countries where you cannot actually go where the problem is. So, and that will go far beyond rabbits. So I think one of the next species on the list could be the European carp, and then it should go to other species, but it's more of a political discussion. I will give you an introduction on the problem. So why do we do something like this? How are we going to do it? And what kind of research can be done kind of in the process? And that's where I'm involved. And that also shows you what former PhD students of mine have done. And here you really need to stop me if you do not understand this, otherwise it's going to be completely over the head, and that would not do the subject justice, I think. Okay, so here we have a rabbit, and I will also explain you later what is the difference between a rabbit and a hare. They are not the same. And they are fairly cute little things. However, in the Australian landscape, they are everything but cute. So they're really responsible for plant and animal species disappearing, and some of the effects will take a long time. So, and the rabbits are particularly dangerous when there is actually not much else around. Then you do not need many rabbits to keep, for example, certain trees from rejuvenating. You won't see the effect in the first 50 or 100 years, but eventually, because you do not get young trees again, the whole species will disappear. Although, for a long time, you've seen quite normal, big, healthy adult trees, but you will not get young ones. So there is a kind of a long-term, almost unexpected effect of having rabbits in the country. On the other hand, the effect in other countries is quite the opposite. So, okay, a little bit about the introduction of the European rabbit. So the European rabbit is native to the Iberian Peninsula, so that's Portugal and Spain in Europe, and it belongs there, and it forms very important functions on that peninsula. So it's a prey for the lynx or the imperial eagle in Europe, and without the rabbit, eagle numbers will dwindle or may even go. The eagle may leave the area altogether. So their function in the ecosystem is quite different from what it is in Australia. Okay, so in 1859, 26 rabbits were released in Victoria by some romantic idiot, if I may say so, who thought like he would like to transform the landscape a little bit, how it looked in England, and rabbit, in his opinion, were kind of a critical part for it. And they were released repeatedly because it wasn't actually that easy to get them breeding and used to the Australian landscape, but eventually it was successful, and quite dramatically so. So in the last 70 years, rabbits colonized most of Australia because the environment is ideal. Rabbits are quite interesting species as far as feeding and digestion goes, so they are fairly unique. So it's a very complicated process, and they are quite efficient, so they are kind of superior to many other grazing species. There are very few predators, and there are even fewer diseases when the rabbits were originally released. So this is a dramatic picture, early last century, and it shows people hunting rabbits and getting money for it. So they are even a valuable commodity, so you can eat them, but the pelts are probably the most important thing, and the head money you get from governmental organizations. So, and they can become a pest species because only 3 months until sexual maturity, and then you get the next generation. Gestation period is only 28 days, litter size 4 to 8. Remating may occur immediately on the same day, within a few minutes, if you like, of the last of the litter being delivered. Rabbit numbers peaked in the 1940s at approximately 0.5 to 10 billion rabbits per Australia. So that's a lot. So you would see in some areas whole carpets of rabbits moving. And if people complain that the biocontrol is not efficient and there are still lots of rabbits around, yes, but nowhere the numbers Australia used to suffer. And the rabbit control by people like this was never to be very efficient because these people caught a lot of rabbits, but they made a living out of catching a lot of rabbits. It's not really in their interest to catch all rabbits. So this kind of helped to manage the problem a bit, but wasn't really a very efficient thing. Okay, and if you want to see what rabbits can do on a landscape scale, you can go to Macquarie Island, and I think things on this island is changing because they have rabbit eradication programs and they can be very successful, i.e. you can kill the very last rabbit on a relatively small island. But this will be probably impossible on a continent. However, here on Macquarie Island, photo taken when there were still lots of rabbits around, and they put a fence around a small area, and this is a particular type of, they call it cabbage, but I'm not sure whether it's actually a cabbage plant. And that is a native plant to the island. This is how the whole island is supposed to look like. And this is how the whole island looks like when there are rabbits around. So they transform the landscape. And you can tell that in this little jungle, it's much easier for the seabirds to breed and to evade predation by other seabirds. Whereas here, everything is in the open. So it has dramatic consequences, not just for this island, also for our landscape. So what was done conventionally to control the rabbits? So there is shooting and trapping, but if you speak of billions of rabbits, there aren't enough bullets to go around, let alone the people who pull the trigger. So you cannot shoot the rabbits into extinction. Similarly, you cannot trap them into extinction. So you can trap the rabbits in your garden, you can trap the rabbits in an enclosure, but you cannot really do this in a national park. So people resorted to fencing, rabbit-proof fences. There's even a movie out called The Rabbit-Proof Fence. It's somewhat effective, but they tend to be damaged by all kinds of things. So large animals damaging them, landslides, all kinds of things. And once breached, then the rabbits are difficult to control again. Warren ripping and fumigating is efficient on a local scale, but also quite difficult if you see that the ANU is heavily infested with rabbits and they tend to breed underneath the building, so that also has limitations there. Then rabbits can be poisoned, but then again, you have to apply a lot of poison and repeatedly so. So what you see here is a driving of rabbits into mine shafts. So, and this is not just like an invention, this actually did happen. So what it indicates really is that there were a lot of rabbits around previously. Okay, so the first biological control agent released in Australia was Myxoma virus. That belongs to the Pox viridae. It's the same family that has our smallpox virus, which no longer exists in the wild. So, and this Myxoma virus was released in the 1950s, and it caused a disease called myxomatosis. Initially, the case fatality rate was up to 99%, but in a few years, due to attenuation of the virus, so the virus became less virulent, so less killing power, if you like. Then also the rabbits became genetically resistant, the outcome the same, and less virulent strains became dominant in the field. This is actually not nice for the rabbits this development, because what we see here, and you can see this occasionally in Canberra as well, is rabbits suffering from myxomatosis, and this one, I guess, is dead now, but it took quite a time to die. And they can waste away the areas around the head, and the eyes in particular. They are heavily swollen. They can't eat properly anymore. They have behavior abnormalities. So they kind of waste away, and that can take weeks. So this is not a swift, humane kill. So things improved a little bit with the release of suitable vectors like the rabbit flea, and now if you catch a wild rabbit or a feral rabbit in Australia, then they're always flea infested, so that helped the spread a little bit. But, very important, and in contrast to caliciviruses, and I come to that later, is that myxoma basically needs a living rabbit to spread from one rabbit to the other. This is also the reason behind why, if you just let it run its course, evolution will produce a virus that makes the rabbit suffer for long. So the rabbit lives longer, so the virus has more time to spread into a new host before the old host is eventually killed or not. The virus doesn't really care about whether they killed the old host, because as long as it keeps spreading, this is the evolutionary advantage. So, in short, the myxoma virus biocontrol agent has lost its edge a little bit. It is still out. It still contributes to the reduction in rabbit numbers, but for more than one reason, it's not ideal. So now we come, and any questions to rabbit myxoma viruses? Now we come to the rabbit hemorrhagic disease virus, abbreviated RHDV, from the family Caliciviridae. So this family contains also human norovirus, which is not really a killer, but highly infectious. And if you ever had an outbreak in your family, it's something to remember. So I can say this with a grin because I am fairly resistant to circulating human norovirus strains. So there is resistance in the human population. So I have to clean up the mess, in other words. Okay, so that was released in 1996 here in Australia, and it causes the rabbit hemorrhagic disease. So it was and still is highly successful because it led to 95% reduction in numbers. The percentage of killing is even higher. This is close to 100%, back in the day and still today. So far, no significant reduction in virulence has been observed because, in this case, transmission occurs, amongst other things, through blowflies, and that works best when the rabbit's actually dead. So here, the evolutionary advantage for the virus is to infect a rabbit, to produce as many virus particles as possible, and this often happens if the virus just goes for it and in speed produces an enormous titer, and then the rabbit dies and then starts to decay and attracts insects, and they feed on it, and then they harbor a lot of viruses in themselves. So the titers are incredible. And then these insects shit all over the place, and then other rabbits eat grass with feces of these blowflies, and here you go, it goes all again. So out of 100 rabbits infected, susceptible rabbits, 97% or more die within 72 hours. So it's not just killing almost all rabbits, it's also killing them super fast. And in the first 24 hours, you won't even see much in terms of disease. And then in the second and third day, yes, you see it, and it's kind of progressive. And then the rabbit eventually dies. So this is kind of, if you like, good for the rabbit because it really, really limits in a sustained way the time they suffer. And if you kill rabbits by the millions, or if not billions, then I think this should be a reasonable thing which biocontrol agent is chosen over the other. Okay, But genetic resistance to IHDV may still evolve, you never know. Very young rabbits are not susceptible to biocontrol agents of the strain IHDV1 and K5. These are the ones that are officially released. We also have circulation of IHDV2, which in many ways is superior to IHDV1 and K5, but it came to Australia by unknown means, and now it's here, but it has not been adopted yet as an official biocontrol agent. So you can, if you are a landowner in South Australia, you can ask for this one, but you cannot ask for this one. Okay, antibodies against non-pathogenic LIGO-viruses protect against IHD. So the people I'm working with at CSIRO, before I got involved, discovered that probably with the original set of viruses that reached Australia, there was already a virus in them, but it was a virus that wasn't a killer. It was more like human norovirus. It affects rabbits, makes them sick a little bit, doesn't kill them, but then leaves immunity behind, and the immunity is cross-protective. So to some degree, an infection with RCV1 protects against infection with RHDV. What's even worse is IHDV may go a little bit in these viruses, but not kill them, but then even boost the immunity. From that point onwards, this rabbit will not die because of an RHDV infection. So all that means that IHDV may actually not get rid of the very last rabbit in Australia, but it may help to manage the problem by reduced numbers. Okay, what we see here is the impact of control measures on rabbit numbers. And we see here a time scale from 1927 to 2011. And what we see here is rabbit abundance. That is kind of a very complicated unit. It's not just sheer numbers, which is, it also takes account that a lot of damage is done when the rabbit numbers are low, but in areas where there is not vegetation. Okay, well, what we see here is it normally goes up and down and up and down, and we have inconsistent rainfall in Australia, and if rainfall is aplenty, then there's lots to eat and rabbit numbers go up, and if there's a drought, rabbit numbers go down with everything else going down. Okay, so then was the introduction of the disease myxomatosis, and that had a severe effect on rabbit numbers, but rabbits and virus adapted, and rabbits again became more of a pest. Then people introduced the rabbit flea, which helped the myxoma virus to spread. Again, good effect for some years, but then rabbit numbers rebounded again. Then we had the release of RHD disease, or RHDV rather, the virus was released. And then we have a stronger reduction. And this one is a bit misleading because it doesn't go up here, so it kind of fluctuates around here. So this is the most sustainable thing so far, rabbit calicivirus as biocontrol agent. So now, that was just the introduction in terms of why it is important and what does it do on a landscape scale. So IHDV is highly pathogenic, so it makes rabbits sick. And this is a rabbit that has just died, basically hours before, or minutes even before the photo was taken. It's a laboratory rabbit, it's called New Zealand White. So these are the kind of, it's not exactly an inbred strain, it's an outbred strain of rabbits that people use in the lab. You can also get it as a pet. So what you see here is basically not much in terms of symptoms. So it looks like a pretty normal rabbit is just dead. But what you see if you look careful is that this weird position the rabbit is in, and that is a kind of a position that indicates liver damage. What you see if you look closer and remove that the rabbit was feeding basically until the last moment, is that it started to bleed, and that is a sign of hemorrhagic fever, and that makes the age here in IHDV. But if you cut the rabbit open and compare it to a normal rabbit, So this is a normal rabbit, and this is a healthy liver. This is a diseased rabbit, and this is a kind of a autumn leaf color type pattern in the liver. So it's paler, it's blotched, it's also swollen. So this is a sign of a diseased liver. What you also see is a lot of bleeding in the lungs. This is normal lung tissue. You remember they collapse if you open it. This really explains why the rabbit died, even if the fur looks all right and the rabbit hasn't really lost much weight and all of this. Okay, so adult rabbits are highly susceptible. 95, 97%, you know how you measure it. Very, very effective. But young rabbits, strangely, are innately resistant, reasons unknown, to IHDV1. Not IHDV2, but IHDV1. Susceptible rabbits usually die in 2 to 3 days after the infection. So if you look what killed them, you see massive hepatic necrosis, liver failure, fever, and hemorrhages. It's very difficult to see early signs of disease. And subacute infections are rare, and if they occur, then you will have a bit of an icterus, weight loss, and prolonged fever. And transmissions are fecal-oral or through arthropods, flies. So, as I told you, IHDV is an RNA virus of the family Calliciviridae, and here we see, for example, the norovirus group and the human norovirus here. They are related to the Lagos group, and here we have the rabbit hemorrhagic disease virus. So that is the virus genome. So it's an RNA virus. It is ready to be translated. So we have at the 5' end not a cap, but a viral protein that functions as a cap. Then we have 2 open reading frames, a big one and a small one. And then we have a poly-A tail, so that looks like an mRNA ready to go. And we have actually the 2 RNAs that should be considered genomic. One is full length and one is subgenomic. And the small one codes for more VP60 and VP10, and these are the structural proteins. So there is actually, I will not test you on this, but this kind of strategy is kind of interesting because normally the structural proteins are at the other end of the viral genomes, so in HCV and others. And this is because if they are on this end of the genome, so they are made all the time, the ribosome starts here and starts translating. And there could something happen, the ribosome falls off, RNA is damaged, and then all these proteins are not made. But the first one is basically always made. So, but if for whatever reason they appear at the end, then the virus may struggle to make enough. So how much polymerase do you really need in comparison to structural proteins of a virus particle? Only a fraction. So to basically make up for this, the virus has another subgenomic RNA that just makes this. So then, again, every single time this is translated, you make BP60, and that's the main thing you need to build virus particles. Okay, so, and then that was a job of my PhD students, and then by extension perhaps mine as well, is to study the proteins and what they do. And what you can do is, if you do not have a cell culture system, and that's a problem with this virus, so they're really good killing rabbits, and there is no shortage of the virus. If you kill one rabbit with the virus, you have so many viruses in that rabbit, you could kill all the rabbits on the planet. So it's no shortage of viruses, but using rabbits for experiments in the lab isn't really nice, it's expensive, and it's very slow. So it's much better to have the virus also going in cell culture. However, this virus, until very recently, kind of refused to do this. So what we did instead is we made an expression vector, this is a plasmid, put the coding sequence, let's say, for P16, P23, the helicase, the polymerase, whatever you like, in it. And to find the protein later, we tagged the protein with the tag. So there are lots of tags. One is the MYC tag, it's a couple of amino acids, and you can buy antibodies against the MYC tag, so then it's easy to find that protein once it is expressed. Then you have here a cell culture dish with rabbit kidney cells, and you kind of put the DNA with your open reading frame into these cells, and then the cells make mRNA, make proteins, and then you use a stack of antibodies and immunofluorescence to basically put a fluorescent molecule through a stack of antibodies to the cells that actually make this protein. That's clear? And then what you see are all the rabbit proteins here. Not the rabbit proteins, all the rabbit calicivirus proteins. And some are in the nucleus, some are in the cytoplasm, and you cannot see how large they are, but you can see where they are. And sometimes it's a bit weird because depending on the cell, there could be more in the cytoplasm, evenly distributed, or they could be exclusively in the cytoplasm, or, you know, it varies. These things, if you start looking, it looks complex. But then we focused on the virus polymerase. And because we got a little inspired by what we saw, that most cells actually, at one stage, express the virus polymerase to the side of the nucleus. Here, this thin line here delineates the position of the nucleus of one cell that was transfected with this plasmid and stained for the virus polymerase. And you see that the virus polymerase is here in this little blotch. And that reminded us of the immunofluorescent staining of a cell organelle called the Golgi. So what we did next is to actually stain for the Golgi, because the Golgi has its own proteins. So here we see 2.5 cells, and we stained the DNA in the cells blue, and we stained the Golgi green. And of course, every cell has a Golgi. So you see the Golgi nicely to one side of the nucleus, and this is a stack of membranes that has all the normal cellular functions. So then, when we put the polymerase in cells, we observed that cells with the polymerase almost always kind of destroy the Golgi. So the Golgi signal then appears all over the cytoplasm and not neatly to one side, as this untransfected cell still shows. So this is basically a real application of double immunofluorescence staining. So and then you can make it even overlay, and then you see that the cells who are positive for the virus polymerase, kind of this weird looking Golgi staining. And then you always think, oh, I saw something, but is it real? Is it reproducible? And we looked into another cell line, HUH7, and we stained the Golgi. Looks very similar. These are human cells, very similar to the rabbit cells. We forced the cells to express the polymerase. Polymerase staining in these cells looks very similar to the polymerase staining in the RK13 cells. Then we looked for the Golgi. The Golgi basically exploded in these cells exactly as it did in those cells. We did this over and over, so it's a phenomenon that is consistent. So then Nadia, very smartly, identified by looking very carefully at the amino acid sequence, a little peptide, there's a little stretch of amino acid in that polymerase. This is a whole polymerase. And this red bit, she identified at being very, very hydrophobic. So this really likes to be in membranes. So then we did a lot of experiments. And then we identified that this hydrophobic motif has a couple of amino acids that are absolutely essential for membrane interaction, but I will spare you the details because that comes with 150 or 200 different immunofluorescence images. But I show you the result of all these experiments. So we found a motif in the caliciviruses that may make this whole protein attach itself to membrane systems. And then we submitted it. And then the reviewer said, oh yeah, this is all very well, but your little motif is, as you show yourself, buried deep into the protein. So I do not really think this is relevant. Rejected. So we said like, maybe it's right. And then we went to a specialist who is into protein dynamics and how they fold and unfold and how they can move. And we told, we have a motif that seems to be based on publicly available data, buried deep into the protein, but at one stage it needs to come out. And he had a look at it and say, oh yeah, this will come out under certain circumstances. So he said, if you put this protein close to a membrane, it will change its shape and it will pull this little finger out and this finger will get stuck into the membrane. And then we had a model in 4 steps, and the first step is a positioning. So there is a hydrophilic charged loop that likes to go to the outside of membranes, but not to the inside of membranes. And then there is a partially exposed hydrophobic lobe that's just lurking under the surface, and that will only come out if the protein is very close to a membrane. So that's this orange one here. And then we have our little particular thing, which is now yellow, not red. And that's a hydrophobic motif that's buried deeply in. But because these things move as a consequence of the protein going close to the membrane, then the finger comes out and sticks itself into the membrane, and then it's stable again. So that was then so attractive to the same journal that they gave us a front cover. So what it shows is, thanks to this reviewer who did not only make the paper better, he also basically single-handedly was the reason why we got it on the front cover. Okay. Now I'll tell you a different story on a different viral protein. So you may remember that I told you that influenza viruses get into the cells through receptor-mediated endocytosis. So they get taken up in an endosome, the endosome pH drops, and then conformational changes happen that make the virus fuse its membrane with the endosome membrane, and then the contents spill out. So what I may have told you is that if the pH in the endosome drops, there is a little channel in the virus particle that transports the protons from the endosome lumen into the inside of the virus particle. And this is required for the changes in the HA protein, which also have a finger that then pokes into the membrane and started the fusion event of the virus membrane with the endosome membrane. So influenza virus has a porin, a protein that forms a pore. So many viruses have it that rely on this kind of thing. We found that Lagovirus genome also has a protein that could be a porin. And another PhD student, analyzed the amino acid sequence and found there are 2 helical domains that are very typical to a viral porin. These 2 domains will not form a pore in a membrane, but if you have more than 1 P23, then you have 4 or better 6 of these, and they can form a little ring and can form a pore. So critical for this is actually dimerization, or even the formation of trimers or tetramers. Like the porins have to come together, they have to like each other. First, we found that all of these caliciviruses have this kind of stretch of amino acids, so that's always reassuring. And then we looked whether we could provide evidence that these proteins actually like each other and form complexes. And then I went back to something I kind of thought of when I was a student, and that was called a nuclear co-translocation assay. And this is nice because it's another example for immunofluorescence. And I think it looks quite pleasing, artistically even. So what you see here is, so now you know how this is done. This is a plasmid. Now we do not express the virus polymerase, we express a different one. It's P23, it's one of the smaller proteins, and we put a tag on it. So the MUC tag is here later stained with a fluorescent reagent that gives you a green fluorescence, okay? So you put this one into a cell, it is translated, and you have all these proteins, and you can stain them green. And what we think is that they form at least dimers. But you know, this is green and this is green. You cannot tell whether these dots are single proteins, dimers, trimers, tetramers. You cannot tell, you know, dot is a dot. So however, what you can do is, you can make a similar construct with the P23, a different tag. This is called now a flag tag. So you can use a different antibody and you can make this glow in a different color. And it contains a little gray bit, and this NLS, it's a nuclear localization signal. So there's a couple of amino acids that kind of force the protein into the nucleus. So this happens, the protein goes to the nucleus, and then it forms this nicely weird pattern in the nucleus. Blue is again just a DNA stain to show you where the nucleus is. So this is omitted here. So here we have just stained for the MUC tag. Here we have stained for the flag tag, and DAPI stains for DNA. So this is great. You have basically the same protein, tagged differently, and one goes to the cytoplasm, and the other one goes to the nucleus because you forced a nuclear localization signal onto it. Is this understood? If you don't understand it, you will not see them. Okay. So now comes, you put both together in one cell. These were in different cells. So you basically do it's called a co-transfection. You put both in one cell. And then, if these P23 proteins like to go together, then there's a fair chance that a P23 with a MUC tag and a P23 with a flag tag go together. And because the flag tag P23 has an NLS, that will then get itself into the nucleus along with the other one, which is not supposed to be there because it does not have an LLS. So what we saw is that you see here, all the green is more or less in the cytoplasm. Here, a large amount of green is exactly where the flag-tagged proteins are. Because you will also get the situation that you have two proteins that have flag tags go together, go in the nucleus, and then they are just red. But the ones that are heterodimers, like with a flag and a MUC tag, then you drag the proteins which you can stain green also into the nucleus at the very same spot where these are. So this shows that yes, they do form at least dimers. So if I look at this as a type of efficiency, I would think they form even more than dimers. Then I had another student, and I will cut this short, who identified a couple of cysteines, and this is a whole coding sequence of P23, and the cysteines are marked here, 1, 2, 3, at position 41, 207, and 212. And these cysteines, they are responsible, once proteins are close together, they often make bridges, and they covalently link the proteins. And we found out that certain cysteines are absolutely required to do this. If you change them, then, so if you substitute them by other amino acids, then the dimerization does no longer occur. So, in short, this is all very tedious work and it's very nitty-gritty molecular biology, but this means that you understand the proteins the virus makes better step by step. And this is all quite tedious because it's all without a cell culture system for the virus. So this is why Cyro got basically close to $1 million to make a cell culture system. And there's a lot of money, and there was a lot of fun doing research, but that does normally not necessarily guarantee success. Just because someone gives you $1 million does not mean you can actually achieve it. So you can figure out whether you can achieve it, but you cannot necessarily achieve it. So we started with that money making organoids. Does anyone know what organoids are? Ever heard of an organoid? Ever heard of artificially produced meat on fancy burgers in, very vogue restaurants, and which will become more popular and probably should? Okay, so this is basically an application of this technology. So you create tissue in cell culture, so you do not need living things anymore. And you can also do this and create artificial organs and then see whether the virus who likes the rabbit but does not like cell culture may like if you give it an organ that's almost as the organ in the rabbit. So, and we first made intestinal organoids. So these are artificial organs in cell culture that, replicate a little bit like the small intestine looks like, with the intestine cells. And you do this by actually getting a bit of a small intestine from a rabbit. And I will do this again next Friday. So we have go out rabbit shooting. So it's really like spotlighting rabbit shooting, not to reduce the rabbit numbers. We don't shoot enough, but to get samples. And then you dissociate the cells, so you get little cells of it. Then you plate it. This is all incredibly expensive because reagents are very expensive. And then you get the stem cells in the tissue to rebuild the tissue in cell culture. And then you get little organoids that are kind of mini intestines in your cell culture. So we did this. It all looks fantastic. It looks pretty much like the real deal. It was great, but the virus didn't like it. So a lot of money spent, virus still didn't replicate it. And we did this first because the non-pathogenic calicivirus like RCV, the one that kind of cross-protects in some areas in Australia against the real one, likes organoids, such as intestine organoids. Okay, but didn't go. So then we said and this was published last year, we do the same for the liver. So we got the liver out of the rabbit, made little mini livers, and even split up the mini livers and made proper monolayer cell cultures out of these liver organoids. And then we looked whether the rabbit likes that, whether the virus likes this. And that was a success. So what you see here is IHDV2 in livers from hares. Hares are rabbit-like, and for whatever reason, IHDV2 really likes hare livers and hare liver organoids. And the signals you see here is the VP60 protein of the virus is stained green, and they are cellular proteins that stained red, and DNA is stained blue. So the little dots here tell you how many cells you have. So every cell, if you see the enlargement, has a little dot in the middle, that's the nucleus. And what you see here, are little islands that are green, and these are islands that express the virus structural proteins. So it's an indication that you successfully infected the cells, and the virus keeps going in the cell and makes a lot of proteins, and you can see them. The good thing is it's happening, so this is basically the first ever photo of true virus replication in cell culture. And the bad thing is it happens only in little islands. So it's not like the whole cell culture is infected, just a couple of cells here, a couple of cells here. So good, but not as good as it should be. And then you do not really trust one assay. What you see to the left, all these columns here, is like a time course. So it's 1 hour after infection, 24 hours after infection, 48 hours after infection, 72 hours after infection. And just for the sake of it, focus on the panels here below. So this is the multiplication of genetic material in organoid cultures from hares. And you see there is a certain amount of RNA, and that's leftover of the stuff you use to infect the cell culture. You can never wash it away completely. But then over time it grows. And it grows from 2 to 6, which doesn't sound much, but it's a log. This is like 2 is 10, 3 is 100, 4 is 1,000, 5 is 10,000, and 6 is a million. So that really goes up. It actually goes up dramatically, exponentially. And this is exactly what you would see if these cells do their job and really make new virus particles. So we established the first ever cell culture system for rabbit caliciviruses, which now allows you to study all kinds of things without torturing laboratory animals, because you can multiply these organoids without killing additional animals. So, and in the meantime, not yet published, we found out that you can make this even better if you add certain stuff in the medium that silences the innate immune system in your cell culture. Then you can add another log, like another 10-fold production of virus particles. So what you can also do is you look into the species specificity. The fact that the rabbit hemorrhagic disease virus is used as a biocontrol agent is it kills rabbits fairly efficiently, but it does not kill anything else. If it would start killing people's cats, people's dogs, farmers' sheep, wow, they would be. I wouldn't be safe anymore, right? So you have to make sure that the virus goes after the rabbit, but does not really go after everything else. And the virus is specific for lagomorphs. That means rabbits in closely related species. So the rabbit and the hare, this is a rabbit, this is a hare. If you ever see a hare is a substantial bigger affair than a rabbit, so they are actually really different, but they are very closely related. So some rabbit hemorrhagic disease viruses, like the IHDV1, killed rabbits, but not hares. IHDV2 kills rabbits and hares, which is good because we also have a problem with hares. So we now tested whether these viruses would multiply in organoids from these 4 species: rabbit, hare, mouse, cat. And IHDV1 can only multiply in rabbit, and this is the same as this virus does, for real, outside, in vivo. And IHDV2 can multiply in rabbit and hare, but not in the other 2, and this is also what IHDV2 does in the wild. So you have to do these kind of experiments to show very skeptical people that your organoids are real and that they reproduce what is expected of them. Okay, so we're getting over time. This is, I think, the second or last slide. You can get involved. So you, for example, can engage with rabbit scan. So you can record rabbit activity, and also, most importantly, if you find a dead rabbit on the road, or even better, off the road. So don't become secondary roadkill by collecting samples. It's not unusual. So then you can ask for a sampling kit to be sent to you, and then you can sample it and get instructions, and then you can set the sample back. It's all FedEx, all paid. And then you can see your little dot popping up on a map. So they will tell you whether this rabbit died of IHD1, IHDV2, or of unknown causes not related to these 2. So yes. Okay, so if it's a healthy rabbit run over by a truck, so that the truck killed it, yes. But you have to remember, if a rabbit is virus infected, it does not may have all his mental faculties proper, so they have a higher chance of actually getting run over by a truck. So it's not always stupid to sample roadkill. But of course, if a rabbit dies off the road in this overstretched position, then this is a better sample if you have a choice. But these people will process any sample you send them. Okay, and credit goes to, and I would like to select these above all others, Nadia and Elena. And these were former PhD students of mine. They both were extremely successful. Nadia got, 8 papers out of her PhD, Elena 6 or 7. Nadia instantly got a job in Sandstone University in the US. So Alabama first, Pennsylvania second. She turned down a job in California. never understand. And then went to Oxford. And Elena straight away got a job at Cyro. So I'm still working with them. And these are other collaborators, so this kind of work is always a big team. So you're never really going to do this on your own. Okay, questions. Last questions in the whole series. I switched the lights back on. And this is also an indication, if you like molecular virology, this is how basically a PhD looks like. But not the whole, all slides, so you can, so these are the highlights. So there's a lot of hard work in between the slides. Any other question you may have? It does not have to be anything to do with rapids. I can. Okay. So the politically correct answer to this question is to remind students of the definition of a final exam. That's not mine. That is a long-standing definition. That means a final exam is supposed to test the content of the unit, full stop. Having said this, I think it is not unreasonable to assume that there will be a certain focus on the second half, i.e. everything that wasn't already assessed in the mid-semester test. But I reserve the right to ask the occasional question on other bits. So this is as good as I can make it. Any other questions?